Microbes, or microorganisms, are living things that are too small to be seen with the naked eye. Now, when you think of microbes, you might think of germs, these organisms that when they get inside your body can make you sick. But did you know that a lot of microbes are not only good for your body, but need to be there?
Ever since you were born, and even right now, millions of these microbes are working all throughout your body to help digest the food you eat and to prevent you from getting sick. Collectively, this community is called the human microbiome. And on today's episode of Here's the Cool Part, we're going to learn about how most of you isn't you.
The human microbiome is located all over your body. There are microbes in your mouth, your nose, your stomach, your intestines, your lungs, your hair, your ears, your esophagus, your skin, and your gastrointestinal tract. In fact, microbes make up almost 60% of the cells in your body. Now, these microbes are a lot smaller than your human cells, which is why you still look like a human. But when it comes to numbers, the cells in your microbiome outnumber your human cells. And that's why you could say that most of you isn't even you. There are so many microbes in your body that putting all of them together will total 1, 2, 3 pints and weigh up to 2.5 pounds. Now, there are 5 main types of microbes in your microbiome. Bacteria and viruses are by far the most common, but you also have fungi, protists, and archaea. Together, these tiny little organisms work together to keep you and your human cells healthy and your body working normally.
Sometimes life just gets you down. Sometimes you feel like you've fallen so far down that you'll never get back up. And in those dark times when it feels like nobody sees you, nobody notices you, nobody cares about you, just remember this. You might be lonely, but I promise you are not alone. You have 39 trillion microbes in your microbiome walking with you every step of the way. 39 trillion microbes that depend on you, that would not survive without you. 39 trillion microbes who care.
A person's microbiome is kind of like a fingerprint, because everyone's is different, with different types, combinations, and abundances of each. Some people might have more microbes than other people. Some people might have more or less of a specific microbe, and other people might have completely different microbe species altogether. The reason why people have different microbiomes is because its composition is based on your location.
Location!
And your diet. The types of microbes in your body are different based on where you live, the parts of the body you're looking at, and the food you eat. Your geographical location, or the place you live, affects your microbiome because different species of microbes are more common in different parts of the world. For example, if you live in Southeast Asia, you may find that your microbiome consists heavily of a species of bacteria called Prevotella. But if you live in North America, you may instead find that your microbiome consists heavily of a species of bacteria called Bacteroides. Your microbiome composition also differs throughout your body, and because of that, different groups of microbes perform different functions. For example, the microbiome of your skin helps protect from pathogens and helps heal wounds, whereas the microbiome in your mouth helps protect your teeth from damage because it forms coatings called biofilms.
Microbes that grow in your mouth, your esophagus, your stomach, and your colon are collectively called the gut microbiome. Now, the gut microbiome is extremely sensitive to the different foods we eat. Every single microbe in your microbiome has its own unique metabolism, meaning that it requires its own special nutrients to grow. So, depending on the different foods we eat, our microbiome responds accordingly. And since nobody eats the same food as you, everyone's microbiome is different.
What's up, dudes? It's me, Chad Bradley, and welcome back to another episode of Act and Chat with me, Chad Bradley. I care about what I eat because it affects my microbiome. For example, I make sure to eat a ton of fiber because fiber is a great source of food for a species of bacteria called lactobacillus. Now, lactobacillus is a super important bacteria for a healthy gut microbiome. I also make sure to avoid a lot of processed meats, so my microbiome won't be full of a species of bacteria called Dysentery vibrio, which has been associated with inflammatory bowel disease. Hope you guys learned something today. This is Chad Bradley telling you to stay buff and stay brainy.
They're sleeping. Now, every baby, including this little guy right here, is sterile in their mother's womb, meaning that they have no microbiome. In fact, babies get their microbiomes right at birth just from being born. The skin microbiome comes from contact with the birth canal, or skin-on-skin contact with primary caregivers, whereas the gut microbiome comes from breast milk during breastfeeding. In fact, scientists have discovered that breast milk has been slowly evolving for millions of years to include more microbes that produce nutrients and even vitamins. Oh, sorry. Whose baby is this?
As you get older, your microbiome will become more complex since you encounter new microbes in the soil, water, plants, animals, and even other people. Importantly, as you get older and your diet diversifies, these new foods you eat will help shape your microbiome by causing different microbes to grow. Let's take a look at this graph.
This blue A over here is called alpha diversity, or the diversity of microbes within an individual. In other words, that's the types and amounts of microbes that are growing within you at a given point in time. Now, as you see, as you get older, alpha diversity increases, meaning that as your environment gets more complex, your microbiome gets more diverse. Now, here's the cool part. This red B we call beta diversity, or diversity of microbes in between individuals. In other words, the diversity in a population or a community. Do you think beta diversity goes up or down as the population gets older? Pause the video and discuss. Now, the interesting thing is that as we get older, beta diversity in a community will decrease, meaning that the community's microbiome will begin to resemble each other's. Now, this makes sense because you and your community likely spend time in the same places, with the same people, or even eat the same food. So what's really, really cool here is that alpha diversity always increases and beta diversity always decreases. How cool is that?
Now here's another cool part. This pattern of diversity was demonstrated by Vange et al. In 2018. People moving from Thailand to the United States had their microbiomes tested over time. And what scientists noticed was that the longer they lived in the US, the more their microbiome resembled those of European Americans who had lived in the US their whole lives.
So, now that we know what our microbiome is and how we get one, let's talk about what our microbiome can actually do for us.
Okay.
Now, this relationship we have with our microbiome is called symbiotic, meaning that we do a lot for our microbes by giving them a place to live and grow and tons of nutrients from our food, but our microbes also help us in various ways. Kind of like these mushrooms over here. Now, mushrooms that grow on trees like this can't produce much nutrients on their own, so they have to rely on the tree to give them those nutrients. But they actually exchange their nutrients and water with the tree to give the tree things that it can't produce on its own. According to Dr. Susan Lynch from the University of San Francisco, this symbiotic relationship we have with our microbiome can be divided into 3 categories: metabolism, biosynthesis, and immunity. Like every other organism on Earth, the microbes inside of us need nutrients to survive. Luckily, they can get those nutrients from the foods we eat. That means that when we eat food, we're not only feeding ourselves, but we're also feeding trillions of microorganisms that, in exchange for nutrients, help us digest the food that we can't fully digest ourselves. Kind of like a virtual organ. Take these potatoes, for example. These potatoes, along with many other plants, contain what are called non-digestible carbohydrates. Now, that means that they're partially resistant to our body's natural digestive enzymes. So, when we eat them, common gut microbes, such as a species of bacteria called Clostridium, play a huge role in breaking them down. When they do, they turn them into short-chain fatty acids, or SCFAs, like acetate, butyrate, and propionate. SCFAs have antioxidant and anti-cancer properties. So, the next time you see your gut microbiome, make sure to thank them for breaking down those plants.
Microbes provide us with things our body doesn't make on our own. or things that we don't make enough of without them. Although we get most of our nutrients from food, some vitamins, hormones, and even brain chemicals called neurotransmitters are actually made by our gut microbiome. I was just at the vitamin store picking up some vitamin K, and it got me thinking. Up to half our daily vitamin K supply is actually made by bacteria in our gut. Now here's the cool part. Since vitamin K is necessary for wound healing and blood clotting, our bodies would actually have a really hard time stopping bleeding after, say, a cut if it wasn't for our microbiome. How cool is that?
Which of the following does not house microbes? Pause the video and discuss. Scientists aren't sure yet if there's a microbiome in your brain, but what they are sure of is that your microbiome can affect your brain. The gut-brain axis hypothesis, say it with me, the gut-brain axis hypothesis, the gut-brain axis hypothesis, the gut-brain axis hypothesis states that the molecules produced in your microbiome can affect how your brain works. For example, bacteria like Streptococcus and Enterococcus produce a molecule called serotonin, also known as the happy hormone. And a bacteria called Lactobacillus delbrueckii produces a neurotransmitter called GABA, or gamma-aminobutyric acid, which can help your brain calm down from a stressful situation. Now, once again, scientists aren't sure how much of these neurotransmitters that are made in your gut can get all the way up to your brain, but what we do know is that people with large-scale microbiome changes often have changes to their mood as well.
Okay.
Now, here's the cool part. Studies have shown that feeding mice bacteria that can produce GABA leads to great reductions in anxiety and depression-like behavior.
Whoa!
The microbiome is a stable community that helps us resist pathogens when they get inside our bodies. They do this through a phenomenon called colonization resistance. And what that means is the mere presence of the microbiome alone is enough to make it harder for infectious microbes to grow once they get inside our bodies. This can happen in a couple different ways. Directly, because our microbiome interacts directly with pathogens, or indirectly, because our microbiome also stimulates human immune cells that then move on to protect us from infectious pathogens. Many different species of bacteria in our microbiome produce peptides with bactericidal properties called bacteriocids. That means a bacteria will release these proteins that then go on to kill other, more harmful bacteria that isn't part of our gut microbiome. Even the viruses of our microbiome, called bacteriophages, help us out by getting inside of weak, infected cells and then breaking them open in a process called lysis. Kind of like a balloon. Even when our microbiome isn't actively killing pathogens, they're still stopping them from growing simply by being there. You see, bacteria and other microbes have to compete for limited nutrients and space. So when we get infected by a pathogen, they might not even be able to grow because there just isn't enough food or space to go around. If we didn't have a microbiome and we got infected by a pathogen, they would have free reign to go wherever they wanted and eat whatever they wanted to eat. The pathogens in your microbiome are a lot like teenagers in a subway. When the subway is full of people, they tend to behave. But when the subway is empty and no one's watching, that's when they can get a little mischievous.
Our microbiome can also protect us indirectly by stimulating our immune system rather than just interacting with pathogens. Immune cells called neutrophils and macrophages are cells in our immune system, and they're essentially like the first responders to a site of infection. Now, in people with healthy microbiomes, studies show that these neutrophils and macrophages have increased levels, and therefore these people can fight infections faster. But in individuals with an unhealthy microbiome, these cells are decreased, and therefore they're more susceptible to infection. Aww. This exact phenomenon was demonstrated in studies with germ-free mice, or in other words, mice without a microbiome. Now, in germ-free mice, immune cell production goes way down, and therefore they're way more susceptible to infections. But here's the cool part. When these mice have their microbiomes reintroduced, immune cell count goes back up. This demonstrates how integral our microbiome is in the development of our immune system. Now, we have to remember that this relationship with our microbiome is a two-way street. Not only do changes in our microbiome affect us, but changes in our genetic makeup can affect our microbiome. Now, when scientists study these changes, they often make use of sets of twins. The first set of twins are monozygotic or identical twins, but they also study dizygotic or fraternal twins. Monozygotic twins, as the name suggests, come from the same zygote, meaning that they come from the same sperm and egg combination. Now, monozygotic twins share close to 100% of their DNA, and that's why they look so similar. Dizygotic twins, on the other hand, come from a different sperm and egg combination, or two zygotes. Now, dizygotic twins share about 50% of their DNA, just like any other sibling. And that's why they look similar, but they're never going to look identical. A study by Dr. Julia Goodrich and Dr. Ruth Lai followed these different kinds of twins and measured their microbiome compositions to see if changes in genetics affected what kind of microbes are present in your gut microbiome. Now, here's the cool part. Monozygotic identical twins had much more similar microbiomes than dizygotic fraternal twins, even if they were eating the same food, going to the same locations, and spending time with the same kinds of people. Now, studies like this shed light onto truly how fascinating the interactions between our genes and our microbiome are. Our genes affect what kind of microbes can grow in our microbiome, and our microbiome, in turn, affects our health. So our genes are still affecting our health, but are modulated through our microbiomes.
A lot of what we've talked about so far, we know thanks to DNA sequencing. You see, DNA is a set of chemical instructions that all of our human cells have that tell our cells how to behave or what to make. Some parts of your DNA get transcribed into messenger RNA, or mRNA, and then that mRNA creates chains of amino acids that then fold into a final protein product. Now, this whole process is called the central dogma of biology, and it also applies to all of the microbes in our microbiome. All bacteria, protists, fungi, and even some viruses have unique DNA codes specific to that organism that give them instructions. And since they're unique, we can use them to identify whether or not there's certain species in a sample. Scientists will sequence the DNA of all of the organisms in our microbiome, and then use that to piece together which species are in a sample and how much of each. Now, they do this through a method called shotgun sequencing. Shotgun sequencing involves breaking apart the genome of an organism and then sequencing those fragments individually. Then, you piece them back together based on where they overlap in order to give you the overall sequence of the genome. The study of genomes and their RNA and protein products are called omics. Now, there are three main omics approaches we use to study the composition and function of the microbiome: metagenomics, metatranscriptomics, and metaproteomics. Metagenomics is the study of all of the DNA in all of the organisms in a sample. In our case, it's the microbes in the microbiome. You see, because these microbes are so tiny and all mixed together, kind of like a soup, they're too hard to separate and sequence individually, so we have to sequence them all together. That's the meta part of metagenomics. And then once all of the microbes in the microbiome are sequenced, they're then sorted and counted, and we use that to figure out how much of each species is in the sample. Metatranscriptomics means sequencing only the parts of the DNA that become RNA. Now, by sequencing only the RNA, you get a better idea of the metabolic activities that the microbiome is performing, because you're only focusing on the genes that will become proteins. Metaproteomics takes this concept one step further. Since there's additional changes between RNA and protein, we still don't have a perfect picture of what the finalized protein products or metabolites of our microbiome look like. Metaproteomics allows us to look at the finalized proteins and then figure out what they do in our bodies and even trace them back to specific species.
If you wanted to see what kind of microbes are growing in your nose, which omics approach would you use? Sometimes the same microbe can be good for you or bad for you depending on the food you eat. Which omics approach would you use if you wanted to see if a specific microbe is producing a useful compound or a toxic one?
So the microbiome helps us out in all these different ways. And we figured a lot of this out by sequencing the genes of the organisms in our microbiome. But how does this apply to human health? Well, remember that our microbiome has a perfect balance of species, and that's why it keeps us healthy. It's a lot like this forest here. This forest has high diversity. There's tons of plants, animals, insects, trees, you name it. A high diversity of species is important because it has resistance to damage. Now, what that means is when something goes wrong in the ecosystem, like if a species goes extinct, there's another similar species that can step up and take its place. Our microbiome composition is the same way. You see, we have a high diversity of species, which means high gene count. And this leads to something called functional redundancy, which is just a fancy way of saying that when something goes wrong, our microbiome will remain stable. But if we have low diversity and something goes wrong, then that can lead to disease. So really, it's all about balance. This imbalance in your microbiome is called dysbiosis. Now, dysbiosis can occur in a lot of different ways. It could be the result of a dietary change where you increase your uptake of sugar or other food additives, or because of poor dental hygiene, which allows bad bacteria to grow in your mouth. Ow! Even high levels of stress and anxiety can imbalance your microbiome. When your microbiome falls out of balance, you could use antibiotics to get rid of all the bad bacteria that's making you sick. But antibiotics also get rid of all the good bacteria in our gut microbiome. And without that balance, that bad bacteria can come back and make you sick again.
Antibiotics, compounds that kill bacteria, are A, good for you, B, bad for you, or C, depends on the situation.
Yeah, so C. diff is an opportunistic pathogen, meaning that it sits in your gut and it waits for an opportunity to grow. This can usually happen after exposure to antibiotics, where there's suddenly space in your gut microbiome for the C. diff bacteria to outgrow your healthy microbes. What we are currently using to treat these infections is what is known as a fecal transplant, where we essentially take a fecal sample from someone with a healthy microbiome and give it to the patient. In 80 to 90% of cases, the bacteria from the fecal sample outgrow the C. diff bacteria and the patient's microbiome goes back to normal. This kind of treatment actually works for a variety of related disorders.
So are there other ways to help people with these kinds of dysbiosis disorders without using a fecal transplant?
Technically, yes, with probiotics and prebiotics. So probiotics are food or medications that contain living strains of bacteria and other microbes that can help restore your gut microbiome from illness or help build up useful microbes to protect against illness. Now, when we talk about prebiotics, these are foods or medication that contain food for your already pre-existing microbiome, helping the good ones outgrow the bad ones. Now, the information that we get from metagenomic analysis is helping us figure out what kind of microbes make up a healthy microbiome. And we can then use this information to essentially custom build probiotics, which is nothing but mixtures of different healthy bacteria for different diseases. It's essentially a more precise version of a fecal transplant. So there are treatments available for Crohn's disease, irritable bowel syndrome, and there's actually research happening right now that is looking at using fecal transplants to help patients with depression, obesity, or even dementia.
So metagenomics is helping us figure out what a healthy microbiome looks like, so we know what kind of microbes are missing and which microbes shouldn't be there.
Exactly. So metagenomics is the way forward for microbiome-related treatments.
Wow, Dave, what an episode that was.
That's right. In fact, let's take a look back at everything we learned. We learned that we have roughly 39 trillion microbes that grow all over and in our bodies, which is a number that is larger than our human cells.
We also learned that our microbiome composition differs from person to person because of where they live and where they eat.
Then we learned that we first obtain our microbiome right at birth through skin-on-skin contact and breastfeeding.
Right, but even though we get our microbiome at birth, it gets more and more complex as we age since we encounter new microbes as we grow.
But even though our individual microbiomes get more complex, the microbiomes in a population start to resemble each other because a single population shares the same location and general diets.
The microbiome is also necessary for human health because it helps metabolize the food we eat, biosynthesizes important proteins and molecules for us, and even helps fight infection alongside our immune system.
Then, we talked about how our genome affects our microbiome, which scientists found out through monozygotic and dizygotic twin studies.
The way we study this microbiome is performed through 3 types of omics fields: metagenomics, metatranscriptomics, and metaproteomics. These methods tell us what species are in a sample and what constitutes a healthy microbiome.
An unhealthy microbiome occurs when there is an imbalance in your natural microbial distribution, which is referred to as dysbiosis.
Lastly, Dr. Harpreet taught us about probiotics and prebiotics and how we can use metagenomics to build custom probiotics to treat disease.
Well, folks, that's all the time we have for today.
Join us next week when we talk about why worms are so darn weird.
But until then, here's the cool part.
For example, if you live in Southeast Asia,
Not South America.
oh my God. For example, so what scientists
Energy!
So, that hurt my knee. Oh, my legs.
Look like you're enjoying it more.
Is your
Scene
mic on?
16.
I don't think your mic's on.
Where's the mic?
Scene 16. Scene 16.
Hey, it's Dave. Can I get a hoya? Hoya?
Oh, dude, careful.
Everything's fine.
Not for long.